I'm Dr. Eleanor Drummond and I'm the Blue Sand Research Fellow at the University of Sydney. I first became interested in Alzheimer's disease when I was a third year neuroscience student, and I went along to a lecture by a professor who gave this really inspiring account of everything that we know about Alzheimer's disease and what we don't know. And it really struck me that there are a lot of gaps in our knowledge, which made me really interested in trying to spend my career studying those gaps and trying to work out what causes the disease. Researching Alzheimer's is really important because around the world there's tens of millions of people with Alzheimer's disease, and in Australia it's actually just come out that it's the leading cause of death of women. So we really need to spend more time researching this disease to work out what causes it, and also to obviously try and come up with a cure, because this is desperately needed. One of the significant knowledge gaps that we have in the Alzheimer's disease field is trying to understand what causes Alzheimer's disease. And while we don't know what that is, we can really gain some interesting insight by looking at those brain regions that are what's called selectively vulnerable. This is where some brain regions are much more likely to die than other surrounding brain regions. And if we can work out why that's the case, then that would be able to tell us some really important things about what may be driving Alzheimer's disease itself. So through the Dementia Australia Research Foundation grant that I was awarded, I've been studying this exact thing. Thanks to the grant from the Dementia Australia Research Foundation, I have been able to look really closely at these vulnerable brain regions, and I am doing so using a really state-of-the-art protein analysis technique called mass spectrometry, which allows me to simultaneously quantify every single protein change that is happening in those brain regions at once, which provides a really nice roadmap into why these brain regions may be selectively vulnerable in Alzheimer's disease. The next important part of my research is to try and work out which one of these many protein differences that are occurring in the selectively vulnerable brain regions are actually involved in causing the disease. And to do this, I use a technique called immunohistochemistry, which is a really nice way for me to visualize directly in human brain tissue where these proteins are located and whether or not they're associated with the neurons that may be dying in Alzheimer's disease, or those neuropathological features in the Alzheimer's brain. My research involves taking cross-sections of human brain tissue and using a technique called laser capture microdissection, which allows me to really precisely microdissect out regions of the brain or even individual neurons from these very vulnerable brain regions. The results from my study will hopefully provide a really nice roadmap of all of the protein changes that are happening in the Alzheimer's disease brain. I'm really hoping to focus in on those changes that happen early in disease, because this is ideally when we want to develop drugs to try and treat the disease. One of the things I enjoy most about my research is finding out something new. Every day I'm looking at my new results and I'm seeing something that no one else has seen before, and I genuinely feel optimistic that this is providing new clues about what's causing Alzheimer's disease. So our recent study that we just published looks at all of those proteins inside of these tangles, and we've actually identified 125 proteins that interact with the protein tau, which is the main protein inside these tangles. I'm really hopeful that my results will really provide us with new ideas about what's causing the disease. Ultimately, my goal in future studies is to follow up on these protein changes and work out which ones of these can be drugged. I want to know which ones of these are potential drug targets and work with medicinal chemists in the future to try and develop new therapeutic approaches. I really want to thank Dementia Australia Research Foundation for making this research possible, because I genuinely couldn't be doing this study without this support.